Hello, and welcome to this VJHemOnc podcast. In this episode, we explore the growing role of artificial intelligence in hematologic oncology. From improving cancer diagnostics and accelerating clinical trials, to enabling more precise risk prediction and shaping the next generation of cellular therapies, AI is rapidly changing how we approach blood cancers. We will hear from leading experts on how these technologies are already being applied in clinical and research settings, as well as the challenges that remain in bringing AI into routine practice. So, let's get started. AI is no longer a future concept. It is already being applied to address some of the most practical challenges of hematologic oncology. One of these is clinical trial enrollment, where identifying eligible patients remains a major bottleneck. Gerds discusses how natural language models can rapidly screen patient populations and dramatically improve trial efficiency.
AI is here. AI is here to stay. It's everywhere. And I think we need to harness its power for good. And one of the ways we can harness its power for good is doing clinical research. One of the major barriers to enrolling patients on clinical trials are actually identifying and pre-screening those patients, particularly patients who have rare diseases. We used polycythemia vera in our abstract, particularly for two reasons. One, it's a rarer disease, and most of the care doesn't happen at main academic centers. It happens at regional facilities, at doctors' offices, away from the main campuses, if you will, of academic centers. And so how do we reach those patients with a trial that's a frontline study where they might benefit from it, or at least we can get them on the study and see if these therapies are of benefit to them in the greater polycythemia vera population? So what we did is we have a partner, Diania Health, who has developed this medically trained natural language model. And we used that model, basically took the trial protocol, plugged it into the natural language model, and used it to look for patients who might be eligible for this clinical trial. And within a few minutes, we were able to identify over 30 patients who were eligible or nearly eligible for the clinical trial, just like that. Right. And so just for a sense of context, I'd be in my clinic normally seeing these patients. I might get a patient once a month or every other month that potentially would be eligible. And then I'd counsel them on the trial. And then I might get a patient on every 3 or 6 months. But just to plug it into a computer, and out spits 30 potential participants for a clinical trial. I mean, that is efficiency at its peak. And so I think this is a really powerful tool that we can use to help do trials more efficiently, more accurately, and get these newer treatments out to patients faster.
These approaches highlight how AI can streamline clinical workflows and improve access to novel therapies, particularly in rare diseases. Beyond clinical trials, AI is also transforming how we diagnose and classify hematologic malignancies. Stephen Ansell explains how AI-based image analysis can rapidly interpret pathology samples, delivering insights in seconds rather than days.
I think this is something I'm really very excited about because I think this is the cusp of the next big thing in lymphomas. So lymphovision is really utilizing a typical H&E stain done on patients' biopsies and specifically using a AI-generated large language model learning way of training, what the image looks like and correlating that with is it lymphoma versus not, and then correlating on is that whether or not the patient has a good outcome or a bad outcome, or also what is cell of origin and is it associated with one picture or image versus another. So right now where lymphovision is effective is taking the H&E stain and say lymphoma versus not lymphoma, and then if lymphoma, identifying about 10 different subtypes of lymphoma, and then on top of that, being able to say in large cell lymphoma, for example, so the cell of origin is GCB versus ABC, and then possibly even the dark zone, double hit, triple hit subtypes. And I think the reason this is very valuable, in seconds, you have this answer compared to the usual many days that it takes to complete the immunohistochemistry and often the FISH analysis. So it's really going to accelerate our ability to bring treatment to patients sooner. And the reason that really matters is many studies have shown those patients that don't get treated or need to get treated right away and can't go on trials are the ones that do the worst. This will help to fix that problem.
By accelerating diagnosis and classification, these tools have the potential to shorten time to treatment and improve patient outcomes. Another key application of AI is in predicting outcomes regarding treatment decisions. Ciara Freeman discusses how AI models can integrate clinical imaging and biomarker data to better predict response to CAR T cell therapy. I'm
really excited to be presenting this work. The goal of this research is very simple. How can we do better in helping us understand upfront which patients are going to do really well and which patients are going to not do so well with CAR T-cell therapy in particular? It's a big deal for patients. It's an investment for them and their families and their time. And so we're really trying to utilize these new techniques that we have available. So artificial intelligence, or AI, is giving us a whole new set of tools to perform really great science. And so by leveraging some, really great AI techniques, we're able to harness the data that we collect from lots of these patients as part of routine care. So not just their treatment history, but all the routine lab investigations that we do as part of our standard of care. And in addition, what we found was if we start with just the clinical information that we have, so the patient's history, how they are, and their routine lab tests, we can really identify groups of patients who've got very different outcomes. But when you layer in on top of that information that we can leverage out of their PET scans, so a measurement called metabolic tumor volume, or MTV, which basically is a sort of global sum of all the areas that are lighting up on the PET scan. If you layer that in, you improve the discriminatory power of the model. And what that means is you improve the way that the model can understand which patients are going to do really well and which patients are not going to do so well. And then if you add into that a serum biomarker that's increasingly available called soluble BCMA. So it basically measures how much of those little bits of BCMA are floating around in the bloodstream. If you layer those three things on together, you really improve the discriminatory potential. And the goal of that is to really help us understand, okay, you are the kind of patient who's going to do incredibly well and have a very good outcome from this CAR T-cell therapy, versus you are the kind of patient who is going to be at high risk of early treatment failure, and therefore maybe we should treat you in a more different way, maybe enhance our monitoring or add in alternative therapies, or think about clinical trial options. We've got all these great trials looking at 2 targeting CARs or, combination therapies. So that's what we're really trying to help understand. All of these information that we use to build a model, it's all being pulled from data that we have before the patient gets treated. So when you still got the opportunity to make a decision, and ideally we're going to try and leverage that to help guide physician decision-making in the future when we feel like we've got something super robust that we can share with the community.
Importantly, these models use data available before treatment begins, opening the door to more personalised and proactive care. Looking ahead, AI is also playing an increasingly important role in the development of new therapies. Zinaida Good highlights how AI can guide the design and optimization of CAR T-cell therapies using patient data.
I'm excited about the possibility of AI changing how we think about developing new T-cell therapies. And specifically, there's some initial proof of concept work that our group has done to show that you can potentially improve CAR T-cell function that will be guided by patient clinical data. In addition, there are exciting opportunities in binder design and protein design for T-cell therapies that some of the AI-based methods unlocking. And another 1 or 2 ways that I wanted to mention is first, AI methods can be really helpful in diagnostics from routine data. For example, diagnostic pathology biopsies or clinical data sets that are health records. So, there's, I think, those types of methods where we use patient data that's routinely available to forecast patient outcomes will be deployed like as one of the first applications. And later in the future, it's possible that some of the AI-based models will help us stratify patients based on which kind of therapy they should have, or potentially even design a therapy for them in the future that we could synthesize really quickly for them in a few decades.
Alongside this, advances in protein engineering are further expanding what is possible in cellular therapy design. Andrea Schmidts discusses how AI-based tools are being used to design novel binding domains, enabling faster and more adaptable CAR T-cell development.
Yeah, my lab in the recent years has been very excited and interesting in leveraging these new AI-based tools that allow us to, in a targeted fashion, design protein binders against target of interest. And we think that this is a very powerful technique because it helps to very much scale up this process of generating new binding moieties that we can then also employ for CAR engineering. Because traditional methods to do this would have been to, for example, immunize a mouse and then kind of check what immune response that mouse has and what antibody it develops against it. And so from there to derive a new binding moiety. And so these new tools that we have dramatically speed up the process at which we get new candidate binders. Of course, we then have to vet them and thoroughly test them. But just, the scale and speed that we're able to generate these new binding domains for us has been just very exciting and something that we have been fully leveraging in the recent years. So the way that we have been employing these AI binders is that we use them as the binding moiety of the CAR, of the chimeric antigen receptor, which is the part that really mediates what the T cell then recognizes and attacks. And traditionally, people have used antibody-derived domains for this purpose. And so what we're doing is that we're basically replacing this with these AI de novo designed binders. And where we think that this is particularly powerful is when we're thinking about cancers under therapeutic pressures changing and modifying the antigens that we're targeting. So we are very excited to specifically use these AI-based binders to design them in a way that they're able to rebind escape variants of these target antigens and or deliberately just broaden the coverage of these antigens that we're going after. Since this is also clinically a major problem that we're dealing with resistances occurring to CAR T-cell therapy because the tumor evolves and tries to escape them. So this is where I think this technology is particularly powerful and helpful. So the work that we're doing is right now at a preclinical level. So meaning that we've designed these domains and we've submitted them to thorough testing. The testing procedure that we do is actually quite comparable to a standard new CAR T-cell product. So meaning that we're doing a couple of in vitro assays to just test the properties, the potency of this new CAR T-cell product that we generated. And we also use them in preclinical mouse models to check their efficiency. And the next step now for us will be to move this into translation and to bring these also to the clinic.
Together, these approaches could help address one of the major challenges in cellular therapy, resistance and antigen escape. However, for AI to reach its full potential, access to high-quality data across institutions is essential. Gianluca Asti introduces federated learning, an approach that enables collaborative model development without sharing sensitive patient data.
Basically, we have been working on a few European projects, and during these projects, we have tested federated learning. Basically, this technology allows us to train artificial intelligence models for diagnosis and prognosis on our data sets without data sharing. And what happens is that we share the model at the end of the training, and this allows us to create a more powerful centralized model between different centers without data sharing. And this is awesome because, of course, it doesn't break any GDPR violation or anything like that. And at a certain point, we wonder what happens if we apply this to generative AI. Usually with these rare hematological diseases, if you want to train a model to generate synthetic patients, you need a lot of patients. So this technology fit very well with that purpose. Basically, we trained 3 different models, a Savai, a Bayesian network, and also a GAN model. And basically, the performance shows that the model created in this federated way creates better patients than the ones that we would have been able to create with the data in each singular institution. And also we used a framework for the validation of this synthetic data. We validated that the data were clinically correct, but also that the privacy of the patient was conserved and it wasn't just a copy-paste, otherwise that would have been, of course, a problem. And so, this is the whole concept, and it was really interesting. We're still working on these European grants, and we're plan to have more results by the end of these grants, but so far these results are really promising.
This type of innovation is critical for rare hematologic diseases, where data availability is often limited. Despite these advances, significant challenges remain in integrating AI into routine clinical practice. Amin Turki discusses the importance of data infrastructure, real-time integration, and validation, in ensuring that AI tools can be safely and effectively deployed.
In my view, there are many opportunities for the patients undergoing allogeneic stem cell transplantation, in particular in the terms of monitoring and risk prediction. We know already from previous studies that there's a strong center effect and that larger centers provide substantially better care to patients. And that's why we try to treat as many patients as possible in centers. And I think this is very well demonstrated, and this is something that we appreciate. However, there's still a certain proportion of patients, for several reasons, that are treated also in smaller institutions and smaller centers. And this can be geographically motivated, such as patients who have a difficulty also to accessing other centers. But this has also have other reasons, such as also funding. And this is not only a US-based problem, but also a problem seen globally, because we have centers of different sizes. And my view is that decision support system, but also patient empowerment techniques can really be helpful in that part. We have experimental devices ongoing for the monitoring of patients. There have been previous publications that have been shown, for instance, that patients who have been continuously monitored can be detected for infectious complications upfront before they develop symptoms or before they develop abnormalities also in the laboratory range that would drive physicians to take action. So this is something where we can have warning and support systems. There are other possibilities also to monitor the patients in order to keep them in a steady health status within this challenging situation after treatment. And this is just in order to summarize what we have on the terms of monitoring and surveillance. For classification of complications, we have been working on a graft versus host disease classificator. There are other groups who are working on the classification of chronic graft versus host disease, even utilizing images of the skin. So what I just want to emphasize is that there's a multitude of works going on also in this domain. For the challenges now speaking, we have a challenge still with data integration, because AI, and I mean well-running AI, is building on a good structure, a very solid hospital data structure, and also on principles of data integration. And although we have the possibilities, we have a common language also to speak between different institutions, we need to have the power of the servers and those institutions ready in order to run the devices. And we need also to have the devices connected with an intimate data structure that is enabling to contact and to derive this information within real time. Because if we have long delays between the accessibility of the data and then also the execution of the models that are predictive, we may have the answers not within the desired time frame that we have. Another issue that we are still experiencing, and that's also why the models often stay at the experimental level, is that they become developed and tested within individual institutions, but not beyond. So we have the risk that those models are overfitted to a specific use case within those institutions and not so well deployable to other centers. Federated learning can be one answer to address this challenge specifically, but the implementation of those models and also the willingness to advance further is still within certain limits. Funding is certainly also one issue, because ultimately those devices, when they want to become implemented in routine care everywhere, need to go through quite rigorous validation process and also accommodation process by the federal agencies such as FDA or EMA. And one solution out of this dilemma in order to accelerate this process is that some institutions, and this being said, also the caveat of having the institutional limitation when you say, well, you just have an institution running its own device, but it has the possibility to use a so-called hospital assumption in order to employ devices that are not publicly available within the research purpose within the own institution.
These challenges highlight that while technology is advancing rapidly, successful implementation will depend on collaboration, infrastructure, and regulatory frameworks. Overall, artificial intelligence is poised to transform hematologic oncology across the entire patient journey. From improving diagnostics and accelerating clinical trials to enabling more precise risk prediction and driving the development of next-generation therapies, AI is already reshaping the field. As data integration improves and these tools become more widely validated, AI has the potential to support more personalised, efficient and accessible care for patients with hematologic malignancies. Thank you for listening. To explore more expert insights and interviews, head over to VJHemOnc.com. Be sure to follow us on X at VJHemOnc to join in the conversation, and subscribe to our podcast on Spotify, Apple or Podbean. Until next time.